Hey, hello, this is Celine Romero again, and today I'm gonna show you to do one very important step, and it is getting the data. For example, in this case, I'm gonna look for a paper we recently published. I had the opportunity to collaborate in this paper, which is exploring to cell variability as function, and from here, we're gonna get some data, the very same data we use in this paper. So in this section in the right, you can look for data availability. And if you click on it, should get you to this place. And this place mentions that details of publicly available data is included in methods section. So we got the methods here, single cell expression data sets. And the one I'm gonna look is for the third case. Let's look. Data was downloaded. The third case from 32. The 32 paper is coming from the single cell analysis defining the continuum of cell state in malignant transformation of polyprogression cancer. And in this paper as well, if we go to data availability, you're gonna see this. And the data was obtained from here. Sequencing data have been deposited in gene expression omnibus, GEO. And if we click on here, it's gonna get us to the data. So the data should be here, and I'm gonna walk through that. But if you wanna explore just GEO, you just type it GEO, gene expression omnibus. It's gonna get this web page, and for example here you can look for something you had in mind. For example, single cell mouse breast cancer. Can be human, can be anything. So it says, oh, there are this many. So we click on it, and for example, if I hit on the first, it's gonna get me the summary of what was there. And the overall design is high resolution spatial transcriptomics data. And in theory, I can use this to pull up the data. For example, I click on the first and go to the bottom, and here is all the data. Usually can use the MTX, the TSV data. But for now, what we're gonna do is head back and go back to Becker. And that paper was published by Becker, the malignant continuum of cancer polyprogression. So we have here all the samples, and what I want to point out here is that we utilize two particular cases. This, and if we look for it, it should be there. Why is not there? I'm grabbing the incorrect one. Third case of study, this one. So I look for it, and there it is. This is one sample. The CRC1 is a cancer sample, and if we hit on it, we should be able, in the button, to take out the MTX, TSV, and this TSV. Those are features. Those are the gene names. Barcodes are the tag names for the cells, and the matrix is the cone matrix. That's it. As easy as that. But fortunately, the only thing we need is to copy those numbers, GEO numbers, in the toolbox. So we're gonna do SEG toolbox. It's gonna open, and after it, I'm gonna do import data, and I'm gonna hit in geoaccession numbers. In theory, I can just copy the sample name and hit OK, and should download it. Meanwhile, I'm gonna stop the video because it's gonna take time. But here in the back, it's getting the MTX and everything to build the object. Gonna stop it now and come back when it's ready. All right, the data is downloaded. Then it gives you three options. The problem here is that there could be genes or features with the same name. So here are three options to handle it. So appending an underscore to repeated gene names. Or maybe you just want to remove keep the first and remove the last, or maybe you would like to merge the two of them and preserve the cells and add up all of them. But here I'm gonna just put method 1, which is an underscore. So this is the first data set. And then here we're gonna do the first thing, come back, and as I mentioned, we downloaded this. We come back here, we make sure that's here. So we're gonna grab this as the sample name, and we're gonna assign that. So what we do is here in this pen, and then we're gonna rename cell type, batch ID, or G name. So we're gonna rename batch ID and OK. And then we put the sample name. And in theory, if we hit in the label cell groups, we can see cell type should be nothing because we haven't determined that. But if we do batch ID, should be the sample name. Great. So that's the first thing. Now, if we want to open a second sample, for example, this other, we look for it, and it's going to be here. I'm going to copy that for the sample name, but also I need to copy this number to download it. So I'm going to go into SEGE toolbox again and open another. There is no problem. We can open another. And we're going to do geosession number and rewrite with the new number. And it's going to take time, and we'll let it download. Again, it's processing here in the button. So we wait for it. After it process, again, we're going to do method 1. And here's the sample. And we're going to go back to the website and grab the sample name and go back to that object. Here, we use the pen, and we're going to do batch ID to change it. OK. And we'll rename it, but I recommend doing underscore just in case. Underscore, underscore. And that's it. And here, what I'm gonna do, we can just disregard this web page where you can look for more data sets. This is just one example that I retrieved from this paper. And so far, so good. The first thing we can do is save a copy of this, so we don't have to download and wait for it because it takes some few minutes. So the first thing is, let me just change my path to here. And sorry, here, and I'm gonna save the math files here. I'm gonna show you a couple things. First, save SCE math file, and we're gonna save it. We can keep it like that, Homo sapiens. And cool, we can close it, and then we can save this other math file. And Homo sapiens should be the other version, and we save it. It's not that, it doesn't take that much time. So far so good. What we can do now, it's to do this. There are two things. You can open individual files, type in the toolbox, import data, and load the math file. So I'll process the first one. I'm gonna process this first one, and I'm gonna show you two things. One is that you can QC this in the first. So wait a second. All right, I just verified what I needed. Here is a sample, raw sample. We have way too many genes, 33,000, which is unreal. So the first thing we're gonna look is here in the view and look for cell QC metrics in violin plots. That's gonna tell how good or bad is the sample. I'm gonna drag it. It was in the other screen. So it shows that around 2,000 genes is required, more or less, and the number of reads per counts, it's around like 1/10⁴, and mitochondrial ratio is of around 7%. So if we keep this and we start doing the QC, we hit on here and do the QC filter basic and do the stringent removal. Okay. and accept changes, it tells that it's removing 23,000 genes and almost 5,000 cells. So from this number to these numbers, this data was little bit hard. It's just reducing from 5,000 to 700 cells. So we accept, and that's what we get. And this is something we have to be careful. For example, I'm gonna say this other just in case. It's gonna be Homo sapiens, but I'm gonna include QC in the end. And then I can do the same process for the other sample. Import mat file. And then I'm gonna look now for this other, open. And again, we're gonna look at the QC matrix here in view, and then cell QC matrix. I have to drag it. Okay, this looks a little better. It's around 4,000 features and counts, it's almost similar. The ratio, mitochondrial ratio is almost similar. But if we do QC in this case, I think it's gonna preserve more cells. So it preserves from 9,000 to 7,000, and from 33,000 genes to 17,000. And that's it. We save it. Right. It's gonna come here, and we put the underscore QC. And here I'm gonna show you something very, very important. The first thing is that if we do SEG toolbox again, import mat file, I can, I think I can open a couple of them at the same time. Let me just double check. Import, simulate. Okay, not this way. So I'm gonna open that one, the QC, and wait a second. I'm sorry for that. Yeah, I just got a little bit mistaken by that. So the thing is that we are going to open again and load the SE mat file. Here, we gonna select the two of them at the same time, this and this, the QC ones. Then it's gonna multiple files selected. After reading each file, data will be merged. So sure, I want it. Here, we're gonna do intersect instead of union. So we're gonna do intersect, and it's gonna intersect the number of genes that are in common, and the number of cells should be the same. If I look at the batch IDs, should display the two samples. So this is perfect. We have the two samples merged. And here, the first thing is gonna be, again, do QC, which will do anything at this point. It's already QC. Oh, it did filter some more. So we accept, and it should be the cleanest version at this point. And then we can start doing the embedding. So we hit on here, UMAP 2D, overwrite, yes, and I want to do all genes in this case. And then we let it compute. All right, it finished, and this is how it looks. Once again, I look at the batches, and this is how they look. They have a batch effect, so we don't know if it is biological batch effect or data-driven batch effect, but we just look at that, and that's what it is. Now, what we're gonna do again, it's do the same, but with the sample that is not QC, with the samples. So we're gonna load the two of them that have not been QC, we merge, we intersect, and we do look again at the QC matrix just to be sure. It's gonna pop up this, and it shows little different behavior than before. Then what we're gonna do here, it's gonna do QC again, but remember, this is from completely raw data merged. We do the QC and accepted changes, and now we end up with way more genes than before. That's something to keep in mind. That might be important, might not be. It's worth trying the two of them. So we look at the batch ID, and we have the two samples. Great. And I'm gonna do the embedding UMAP 2D. Overwrite all genes, and I'm gonna stop the video meanwhile. Alright, this is what we got, and as you can see, I think we have some few more cells, and genes definitely we have way more. So what we're gonna do here, it's gonna be comparing the two of them. We preserve 400 more cells than before, and several more genes. So the question is, which one is the best? That's a problem we have all the time. So we have to explore the two venues and decide. In theory, the left-hand side one looks cleaner because it's more dense, and the right-hand side looks more spread. But here's one trick. If I do again the embedding with now the highly variable genes, using existing embedding, no. UMAP 2D, overwrite yes, but now 2000 highly variable genes. Gonna stop the video and come back. Alright, now that's how it looks. It changed it quite a bit, right? So now we're gonna do the same, but with this left-hand side sample. So we're gonna, using the existing no, I want to do the UMAP 2D, overwrite yes, and the 2000 highly variable genes. I'm gonna stop it for now. Alright, and that's what we get. Now, if I bring it on again, the two of them look a little bit more similar. I mean, those are more spread. The difference is that we have some more cells in one sample than in the other. So still, I believe that most of the data is preserved, it's just a different representation, visual representation. And the difference also is some few less 100 cells. That's to be noted. Alright, now what we're gonna do next, we're gonna look at the batch ID, and the batch ID of the other should be similar, and I'm gonna do the clustering, which is here. So we're gonna do clustering, and it's gonna be over the UMAP 2D. It's gonna be the k-means, and let it be, instead of 80, I don't expect like 80 different cell types, but maybe 40. So I'm gonna do 40. 40 is because it's gonna partition in 40 clusters. Similarly, gonna do here, cluster cells, yes, UMAP 2D, k-means, and again, I'm gonna do 40. And that's it. That is what it is. And for last, I'm gonna assign cell types. Cell type. What is this? This is human samples. And yes, automatically. And dynamically, you can see how it's being assigned to cell types. I'm gonna stop it for a few seconds. After it finishes, it's gonna ask if you want to merge them, and that's what we have. And let's look here in the right hand side. Likewise, we're gonna do here, assign cell type to groups. Okay, it's human, yes, automatically, and I'm gonna stop the video and come back when it's finished. After it finishes, it's gonna ask if we merge. So we hit on yes, and that's what I have now. if we look carefully, now here, obviously in the left hand side, I have enterocytes, which is similar here. Great, that's what I expected. Here I have B cells. Okay, fine, endothelial cells here as well. But here identified neurons, and I don't have neurons here except here. I'm sorry. Here. Oh my god. It's gonna be here. So B cells, and these are neurons, like these ones. It says it has podocytes here and enterocytes here. In this data set, we know it's gut, and those are large intestine, and enterocytes or epithelial cells are there. So we are expecting big bunch of enterocytes. Here we have enterocytes, here we have enterocytes, and here neurons. So something is happening within here, neurons, and here these other neurons. But in the right hand side, as you might see, we have fibroblasts that we have also here in the left, and we have macrophages, and I don't see macrophages in the left hand side. So that's something we gotta be careful when handling with the data, that we feel comfy that we keep as much as we need. And that's it. And for finalizing, I'm gonna save this math file, and it's gonna be I'm gonna call it CRC underscore merge it samples no QC. And this other in the left, I'm gonna save it as well as SE math file. And again, it's gonna be CRC underscore merge it samples, and that's it. I should have placed it QZ, but here is the two perspectives, and that's it for this video, and I hope it helps you understand. And later I'm gonna put effort into now what's next, analyzing the data. But this is this is the data set we're gonna utilize. Thank you for watching. See you later.